I got a private message about the proctored quiz. Can I reopen it? Yes, I'll reopen it. I just want to make sure everyone's good on they are squared away for using Proctorio. Now, there we go. Full screen. This is our last bit of new material, and I've been kind of debating basically all morning um, where I should stop, and if I should stop right here. I think what I'm going to do is I'm going to stop at the polymerase chain reaction. That's slide number 10. And so then we're going to reserve 11 through 13, those last three, 11, 12, 13, those last three slides. We're going to reserve those for after our first exam. So the final slide for this upcoming exam is going to be slide number 10 on this PowerPoint. And that's just because it tends to get a little bit the last few slides it gets complicated, and I don't want that to be it gets complicated, and it's the sort of thing that people like to say, oh, okay, well, I need to really focus in on this. I need to focus on my weaknesses because this is a weakness and I can improve my weaknesses and then I'll do well in the exam. And I don't think that's a very good strategy. I think you should play to your strengths and really enhance those. And so this is something that I'm gonna push back on. So I'll stop at the end of slide number 10, and then I'll talk about the exam. So with that said, if some of y'all have taken a molecular biology class or a cell biology class, there's some of this material that might be a little bit of a review. But we're going to take the approach as if it's new material for everyone. So this is all about nucleic acid techniques, and some of these you've probably heard of even outside of an academic setting, outside of class. In particular, like a PCR test for your or PCR COVID test. Those are the sensitive ones. Nucleic acids are great because they can be manipulated. Nucleic acid fragments are generally very easy to work with since they can be all of these things. They can be, there we go. They can be sequenced, they can be duplicated, they can be modified, and they can be expressed. Now, these three things, these three these three techniques are all, well, they can be done using the same instrumentation for PCR. You can sequence a piece of DNA using the instrument that you use for PCR, known as a thermal cycler. You can duplicate it, that's your process of PCR, and then you can modify it also using a thermal cycler. Whenever we talk about expressing, generally we're talking about making a recombinant piece of DNA and basically putting into E. coli and amplifying whatever that product is. Many times it is easier to get the protein sequence information from an organism's DNA than it is from a purified protein. So this, in essence, is basically saying it's easier to work with DNA. I'm going to say DNA greater than protein. And one of the reasons for that is if you think about a DNA molecule, it's a double helix. It's a double helix that is inherently stable. It's different from an RNA molecule, which is typically a single strand or partially single-stranded molecule that can be easily hydrolyzed. Go ahead and add to the chat a thumbs up if you have ever seen the movie Jurassic Park. And I'm gonna try to be an original, or yeah, an original person and say, okay, Jurassic Park 1, not Jurassic World, or okay, good. The central part of that movie is that there's DNA recovered from mosquitoes that are trapped in amber, and it's 65 million years old. Now, there's some validity to that, because DNA is very, very stable. Now, of course, in that movie, the idea is that it's trapped inside a mosquito. And so there's a layer of protection, and then it's trapped inside this amber, providing another layer of protection. If you were to take DNA and basically just expose it to the environment, it would still, like, if you had a solution that had DNA in it, no proteins or anything else like that, if you just had DNA in a solution and you put it in a solution on in a, humid enough environment for preservation, um, and you left it for 6 weeks and you came back to it, it's fine. If you left it for 12 weeks, it's fine. I think that you'd probably be looking at like a year or two before it begins breaking down. So DNA is very stable. Proteins and RNA considerably less stable. So we will talk a little bit more whenever we get to proteins about protein sequencing because it's still a valid process and it's still kind of an essential like tool to have in your tool belt. But it's just when you're trying to find out what is the sequence of a protein, you want to start with the DNA sequence. Because DNA, what we can do is we can cleave it into specific fragments. We can sequence those individual fragments and we can put those fragments back together. So I always think about a double-stranded piece of DNA. Here's my double strand. Almost like a puzzle. So I'm going to go ahead and change color so I can modify this molecule. So let's say that we have the different colors here represent different sequences. So what I could do with the DNA molecule is I could actually take this one right here and manipulate it so that I've taken that blue fragment and I've placed it in between, in the middle of my red fragment. So this requires me to basically cut and paste my DNA. And that's all something that you can do. And the way that you do that is with enzymes or proteins known as restriction enzymes. Now, restriction enzymes cleave double-stranded DNA. They have primarily been isolated from bacteria, and they are not exclusively purified from bacteria, but they're predominantly purified from bacteria. They can be modified, and there are some synthetic restriction enzymes or restriction endonucleases. There are 3 types that you need to be familiar with. And these types all relate to what they're cutting, so what they're cleaving. Now, with that in mind, here's my double-stranded piece of DNA. So those red lines correspond to my phosphodiester backbone. The blue lines here represent my nitrogenous base pairs. Now add to the chat somebody, what type of bond holds my nitrogenous base pairs together? So when you have like G and C and A and T, what type of bond holds those together? Absolutely, it's a hydrogen bond. You're 100% correct. So hydrogen bonds hold they hold the middle together. But what about the red lines here and here? What type of bond hold those together? And you can speak very generally, or you can talk about the specific. There is one specific name for that bond. But if you want to speak more generally, Kate, that's absolutely right. It's a phosphodiester linkages. So these phosphodiester linkages are examples of covalent bonds. Now, what restriction enzymes do is they break phosphodiester bonds. Now, I say that and I want to lead with this because we're going to get into some of the nuances of that. But the 3 types of restriction enzymes, well, there are, and this is one instance where I'm just like, man, biologist and biochemist and chemist, they're the best because creativity is not necessarily their forte when it comes to naming. When it comes to experimentation, oh my gosh, creativity is out of their minds. But, I always like to look at this and I'm like, I imagine a bunch of scientists sitting in a room and they're like, well, we've identified 3 different types. What should we call them? Well, type 1, type 2, and type 3. Simplest explanation, simplest option. So type 1 restriction endonucleases or restriction enzymes, and I use those two terms, endonuclease and enzymes. In this case, I use them interchangeably. Type 1s require ATP, and they have non-specific cleavage. What that means is that when they're looking at a sequence, they don't care. They don't care if they see A, T, C, G, or if they see A, T, T, A. These type 1 restriction enzymes are going to say, oh, there's a double-stranded piece of DNA. I'm going to cut that. And then I'm going to cut here. And then I'm going to cut here. I don't care about the sequence. Type 2, by contrast, don't require ATP, and they have very specific cleavage patterns. So what that means is that there are different restriction enzymes that they'll see, they might see something, they might see a pattern like this, GAATTC. And there's one restriction enzyme that will see this and be like, okay, cool, I'm going to cut right here. Another restriction enzyme is going to see this same pattern and say, I have no interest in cutting there. They're both type 2s because they both have very specific patterns, but that's where they kind of differentiate from one another. Now, they don't have an ATP requirement, which brings us to type 3, which, if you think about these two variables, requirement of ATP, yes or no, and specific cleavage, yes or no. So our type 3 is going to require ATP and have specific cleavage. So it's almost like type 1, type 2, and type 3, the highest, I guess, standard or the highest expectation, I guess, might be your type 3s because they require ATP and they require a specific cut site. Now, with that in mind, everybody listen for just 1 second. Well, listen for the next, 38 minutes. But the next few words are very important. I do not expect you to remember specific cut sites. That is not an expectation. And in the next slide, you'll see why. Here are different restriction cut sites. So if you look at this column on the far left, these are the enzymes. If you look at this column right here, these are restriction or recognition sequences. And then on the far right, the organism that they come from. So let's go over to our first column, enzymes. So all of these right here are the names for different restriction enzymes. Now these names, they kind of seem like a hobbled together mess of letters and numbers. This one right here that I just drew an arrow to, this is commonly known as EcoR1. Some people will look at it and they'll read it and they'll say E. cori. It's EcoR1. This one right here, the one right below it, EcoR2. The one below that, EcoR5. Now, so there's an EcoR1, EcoR2, is there an EcoR3? What about an EcoR4? Or do we just, randomly jump from 2 to 5? There is an EcoR3. There is an EcoR4. This is showing more common restriction enzymes. Okay, now I want to go from this column right here, the enzyme column, to the microorganism column. So when you see that ECO, notice that it's italicized. And if you've taken a genetics class or a microbiology class, when you italicize something, it's either an indicator of a gene or a microorganism. In this case, this ECO is indicating the microorganism that this restriction enzyme was purified from, or where it was discovered. Escherichia coli. E. coli is where EcoR1 was originally found. Now, if you look at this RY13, R25, these are individual E. coli strains. You don't have to worry, you don't have to focus about that or anything. But I always like to highlight this because if you think about a bacterial cell, do bacteria do they have a defense system? They don't have an immune system. They're single-celled organisms. Now, with that in mind, do they have is there anything that poses a threat to bacteria? Is there a biological, or is there a biological entity, I'll say, that basically poses a threat to bacteria? I'll give you a hint and add it to the chat. A virus. Okay. Specifically, what's the name of a virus that can attack a bacteria? Like a bacteriophage. Perfect. Great job. Okay, I'm sorry. I need to go back. I got a question. Why are restriction enzymes necessary? Why do we need to cleave DNA sequences? Well, what I was trying to show you here was this right here. This gives us the power to basically make genetically modified or modify genes. So that's the reason. Okay, so back to where we were. So when you think about a bacteriophage is either RNA or DNA. And these bacteriophage, well, they have specific sequences found within them. Essentially, what EcoR1 is, this is from like an evolutionary standpoint. What EcoR1 is a restriction enzyme that almost served as like a defense mechanism. Because what it would do is anytime it would see a double-stranded piece of DNA, and if it had, starting from the 5' end, GAATTC, and then we've got a reverse complement on our other strand, GAATTC. Whenever E. coli, no, this is absolutely natural. That's what I'm explaining. I got a private message that said, oh, so it's not natural. We're doing it in labs. No, this is absolutely natural. That's what I'm describing right now, that E. coli can do this. We as scientists, and we've kind of co-opted this technology or co-opted this to use it for manipulation. Okay, so with that, if E. coli sees this sequence, what it's going to do is it's going to cut between the G and the A on this strand, and then it's going to cut between the G and the A on this strand. So it's going to be breaking. Now add to the chat, what type of bond is it that it's breaking? And we're breaking. Okay, sorry, it's not a hydrogen bond because it's a covalent bond. It's that phosphodiester bond. Because if we look right here, where I've written my G's and A's, that's where the hydrogen bonds are. So we got hydrogen bonds here, here, here. That's where I've got my hydrogen bonds. So I've cut the strand of DNA, the backbone, the phosphodiester backbone, or the sugar phosphate backbone. So it's those phosphodiester linkages that are broken by these different enzymes. Now, what that means is, and I want you to pay attention to this real quick, restriction enzymes break phosphodiester linkages. Now, the kind of consequence of that is, well, what's going to happen here is we've got this molecule that's, hundreds of base pairs long. And I always like to think about phosphodiester linkages as like the really strong bonds within a DNA molecule. These hydrogen bonds right here, essentially what they are is they're kind of like, they're not as well, you lose the structural integrity of this molecule. So what's going to happen is we're going to end up with products like this, GCTTA. And then here's another product, AATTCG. So those are the two products that are going to result from this. Now, like I said, I do not expect you to know these recognition sequences, but what I want you to do here is look at them and, go ahead and add to the chat how many base pairs are most of these recognition sequences. What's the length of most of these recognition sequences? How many base pairs? Roughly 6. There's some that are 4, there's some that are longer, but they're mostly 6 base pairs. Now, these type 2 restriction endonucleases, those are the ones that we're interested in. Now, if we look at EcoR1, what we've got here is the restriction site for EcoR1. It is going to break this phosphodiester linkage and this phosphodiester linkage. What that's going to do is destabilize that molecule. It's breaking phosphodiester linkages. Then if we compare that to EcoR5, that's going to break this phosphodiester linkage and this phosphodiester linkage. Now, the products of this are going to be different because one of the things that I want you to recognize is that you have one enzyme, which is going to produce, it's going to cut phosphodiester linkages that are not directly across from one another. And then another restriction enzyme where our linkages are directly across. So I'm going to say not across, and the other that are across. Now, these recognition sites in double-stranded DNA usually have what's known as a two-fold axis of symmetry. They're palindromic. What that means is that if you read one way and the other the opposite way, they're going to be basically the opposite of one, or they're going to be identical. Now, enzymes recognize usually 4 or 6 bases. These type 2 restriction enzymes do not have an ATP requirement. And what they're going to produce is, just imagine the separation between this green fragment and this blue fragment. What that means is that these base pairs, they're going to be able to hydrogen bond with their environment, because I always visualize it as like my index and ring fingers. I'm cutting here and here respectively, and then my fingers kind of drift. So then this is an overhang and this is an overhang, and that's the term that we like to use. To visualize that a little bit more, here's what we get, where we've got our green strand, and then we've got our purple strand. This would be analogous to the green and blue strands that are depicted right here. Now, when we have those overhangs, so I want you to these are known as 5' overhangs. These 5' overhangs leave what are known as sticky ends. Alternatively, so this would be a result from EcoR1. EcoR5, on the other hand, go ahead and add to the chat, does EcoR5 generate any overhangs? It doesn't. Does anyone have is there anyone who, and you can send me a private message, does anyone not understand what I'm saying whenever I say a 5' overhang? Okay. So another way that just to really make this point, the way that I always kind of think about it is it's almost like you've got zigzag scissors. Has anyone ever used those crafty scissors that have like a pattern? They give you a cut pattern, as opposed to the traditional straight scissors. So these zigzag scissors would be similar to making an overhang, or a piece of DNA with an overhang. What's going to match up with that? Well, only a piece of paper that also has that exact same overhang. Whereas straight scissors, those would be something like this. EcoR5. The products of this are going to look like such. There's just a gap in there. These are known as blunt ends. I always think that that just makes sense because it's blunted. A piece of DNA has this blunt, abrupt end, whereas the overhang, including strands, are sticky ends. Now, what we do with this is if you've taken a genetics class, I think it's in genetics, it was for me. Okay, so Priscilla, so RB Eco R5 would be a 5' blunt. You don't, it's not necessary to say 5', but yes, it's a blunted end. Great question. Great question. Great job. If you've taken a genetics class, you probably talked about DNA gel electrophoresis. Now, in DNA gel electrophoresis, you maybe didn't run a gel that looks like this. You probably ran a horizontal gel. Now, you can run vertical gels for analysis of nucleotides and nucleic acids. It's a little bit more typical or more common to run a DNA gel that's horizontal. But one of the things that's important to know about this is that you load your DNA sample here, 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 and then you also load what's known as a molecular weight marker or a DNA ladder. Now, that DNA ladder is super important because what it allows you to do is compare the pieces of DNA that you've run to the pieces of DNA that are in your ladder. So if we look at column A here, this would be a DNA ladder. And let's say that this is this highest band right here. This is a piece of DNA that's 1,000 base pairs. Let's say that this is 750 base pairs. This is 600, and this is, I'll go with this one as my lowest one, or yeah, 75, and then this one is 5 base pairs. When you look at those different fragments, that is your way of making a prediction about the sizes of other pieces of DNA. You can say, okay, well, if this top band is 1 kilobase pair, the one below that is 750, then this one right here, well, it's smaller than 1,000 base pairs and it's larger than 750. So let's go ahead and call that 825. Now, if you take a molecular biology class, you will make the concrete analysis where you will measure the precise distance from here to here. And you'll say this migrated, 16.4 millimeters. This migrated 22.4 millimeters based on, and then you make a regression curve or a graph where you say based on these migration distances and the size of those, then this one, because it migrated this other distance, it must be exactly this size. Okay, now that's not something that we're going to get into here, but the migration distance tells you approximately the size of the molecule. Now, if we proceed from here, this is something that's definitely you go into in molecular biology. This is something known as plasmid mapping, or mapping and cutting pieces of DNA, getting restriction fragments. The context for this is if you think back like a generation ago, let's imagine that you have a piece of DNA. And this is a gene that you, like in the 1970s, if you could sequence the gene or sequence a gene, that was adequate work to basically award you a PhD. You sequence a gene. Now, if you can sequence a gene, it doesn't carry that same weight because the technology to sequence genes is so much more advanced. Now, let's imagine that you have this piece of DNA. Here's your 5' end, here's the 3' end, 5', 3'. And if you run this on a DNA gel, here's our DNA gel right here. You've got ladders of known molecular weights. Okay, that's helpful. And then you run this piece of DNA where you haven't cut it or you haven't done anything. Let's say that this is 1,000 base pairs. This is 750. This is 500. This is 200, then 50, and 25 base pairs. Let's say that you take this sample, which I'm going to label it A. A has no digestion. You have not added any restriction enzyme to it. And you run sample A on a gel, and you get a single band right there. Go ahead and add to the chat what you have learned about that piece of DNA, about sample A. And let me rephrase my question, make it a little bit more targeted. Do you know the size of that piece of DNA? Large, somewhat negative charge, molecular weight at 750 base pairs. Yes. We know that it's 750 base pairs because we've got our ladder, our molecular weight ladder in this lane right here, in this column right here. Okay. Now let's say that you take you've got hundreds of copies of this, and then what you do is you take your gene plus eco R1. So you get the restriction enzyme eco R1, and I'm going to use red. I'm going to label this as B. No, I'm just going to, when you mix it with eco R1, you get this and this. First question is, approximately how large are those two bands? Rough approximation. I mean, we're looking at it on a screen here, so you don't have the physical gel or anything like that. But roughly how large are those two bands? 500 and 250. Okay, that's great. So based on them being 500 and 250, does EcoR1 cut this gene? That's not intended to be like a trick question. I think it's pretty reasonable to say, well, the only variable between A and B is the presence of EcoR1. So, yeah, okay. Now, in terms of a sequence, if you, like, again, you don't know anything about the sequence of this gene. You do, however, know that EcoR1 looks for, it's a type 2 restriction enzyme, and it looks for GAA TTC. And when it finds GAA TTC, it cuts there. So you know that your piece of DNA, and we're going to keep a tally up here, from lane A, you learned that your gene was 750 base pairs. Great. The next thing that you learned was that EcoR1 cuts it. If your goal and ambition is to figure out the entire DNA sequence for this piece of DNA, that means you need to figure out basically where, or you need to figure out the identity of 750 base pairs. Now, after running this right here, have we learned the identity of any of those base pairs? Yes, we have. Arguably, after running this, we now only have to learn 744 base pairs. Because we know that EcoR1 cuts it with GAA TTC. So anywhere that there is GAA-TTC, EcoR1 is going to cut it. And based on these two bands, we can conclude that, well, GAA-TTC is in there one time. Now, let's go ahead and use another enzyme. BC gene plus, let's go with BAM-H1. Erase that. BAM-H1. I'm going to jog back a little bit. Got to get our sequence for BAM-H1. That's GGA-TCC. GGA-TCC. Okay, so we've got C, our third trial, where we take our gene that we have now determined six of our nucleotides in the sequence. And now what we get is this, and then we get a slightly lower band here, and then we get this right here. Would anyone add the approximate sizes of those three bands? 500, 250. Perfect. So now what we've done is we've said, okay, well, we know that since our linear piece of DNA gets cut to produce 3 fragments, we know that GGA TCC is somewhere in there 2 times. Because if we look at, it's linear. If we cut it 1 time, then we get 2 pieces. If we cut it 2 times, we get 3 pieces. By us seeing 3 pieces, that means we're cutting it 2 times. So what that means is that we've got our third kind of piece of data, that BAM-H1 cuts it 2 times. So what we can add to this is that we know that GAA, right, GAA, TTC, let me use the eraser, TTC is in there. So is GGA, TCC. And again, GGA, TCC. So we've gone from, all right, we know it's 750 base pairs to, well, we identified 6 of them, and then we identified 12 more. So we are down to knowing the sequence or the presence of 732 base pairs. Now, this is a meticulous process, and this is the technology or the sequencing technique and approach that people would take, 50 years ago. This is no longer the case, but essentially what you are able to do is cut a piece of DNA and say, okay, well, based on this cut pattern, I know this piece of information. I know that this fragment or this particular sequence is present in my gene. Now, this is something that gets very, very complicated because you do a digestion with just EcoR1, and then you do a digestion with EcoR1 and BAM-H1. And depending on the results of that, you're able to kind of piece your DNA back together and visualize, okay, well, based on this banding pattern, this is where these different sites are. So that's the process of restriction mapping, where you basically take a piece of DNA and so are you saying because there are 3 lines on the gel for restriction enzyme C, we can recognize that BAM-H1 cuts it twice, giving us the 3 pieces of... Yes, that's what we're kind of concluding. Because we knew we were working with a linear piece of DNA. And then we add BAM-H1 to it. And because it's linear, I always, I like to get this is my piece of paper. This is my piece of DNA. If it gets cut one time, there are two pieces. If it gets cut two times, then there are three pieces. Now, this might sound kind of trivial, but there are pieces of DNA, or there are molecules of DNA that exist as plasmids, as circular pieces of DNA. Now, if you have a circular piece of DNA and you cut it one time, you're going to see one band. Whereas a linear piece of DNA, if you cut it one time, you're going to get two fragments. So that's an important distinction. But again, this is something that you're going to dive deeper into in a molecular biology class. For this class, what I want you to know is I want you to know that restriction fragments, or identifying restriction fragments, gives you a little bit of information about the sequence. That's what I want you to take away from this. Now, this is just an example where we look at hind 3 cutting a piece of DNA one time. Well, when it cuts this, what do you know about if it cuts it one time? You know that it's linear because you get two fragments. And those two things come together to make something that's 4 kilobase pairs. BAMH1 cuts it two times, and that will give you 3 fragments. Then we do what's known as a double digest, where you do BAMH1 and hind 3. And what that allows you to do, this combination digestion right here, it allows you to say, okay, here's my linear piece of DNA. And I know that based on this banding pattern, my first cut is BAMH1, then there's a hind 3, then there's a BAMH1 cut. So they're all kind of, it helps you identify the sequence and the pattern of them. Now, what this technology has been useful for, in addition to figuring out sequence, is it's been useful for construction and cloning of recombinant DNA. So anytime that, exactly, a vector. So anytime that you manipulate a piece of DNA and overexpress a protein and purify a protein or characterize a protein, you're having to use restriction fragments and restriction enzymes to do this. So restriction enzymes are also useful for mapping of DNA and DNA fragments. So assuming random distribution and equimolar proportions of those 4 nucleotides. This is one sort of calculation that I always like to fall back on and reference is most of our restriction sequences were 4 or 6 nucleotides long. What does that mean for the overall length or the frequency of how often are you going to find something? So I always like to think about, you've randomly generated a piece of DNA. What are the chances that we'll just call this the first position. Like, you've randomly generated a piece of DNA, and what are the chances that in one space you're going to find A? Well, it's 1 in 4. There's a 1 in 4 chance that you're going to find an A. Now, let's just imagine this is 5' end. What are the chances that in the next location, right next to that A, you find a T? And this is randomly generated. So does the A in the first position have any bearing on the T in the second position? Doesn't. This is like flipping a coin. So you're absolutely right. No, it does not. What are the chances that you find a T in that second location? 1 out of 4. Okay. So now let's take this simply. What are the chances that in any piece of DNA, you're going to find A followed by T? Well, this would be 1 out of 4 times 1 out of 4, 1 out of 16. So you could rephrase this as once every basically 16 base pairs, you'll find A followed by T. Does that make sense? Now let's go ahead and imagine our third position. A. It's again 1 out of 4. Then 4th, 5th, 6th. In the fourth position, you got, let's say that you want a C, or you find a C there, then a G, and then a C. In each position, there's a 1 out of 4. 1 out of 4, 1 out of 4. So this is effectively 1 out of 4 to the 6th, which is the same as 1 out of 4096. So one of the questions that I always like to ask is, what are the chances in a piece of DNA that's, or how frequently will you find AGCT in a randomly generated piece of DNA? Or what are the chances, just to use the same numbers, what are the chances that in a randomly generated piece of DNA, you're going to find GGA-TCC? Roughly once every 4,096 base pairs. So if you have a piece of DNA, a randomly generated piece of DNA that's 4,096 base pairs, then you're going to probably find that sequence one time. If you have a piece of DNA that's 2,000 base pairs, your odds of finding GGA-TCC in this randomly generated piece of DNA is considerably lower. So when we talk about these restriction fragments, these restriction sites, they're not super common. They're only as common as the length of them. So if you have something that's, 8 base pairs or 10 base pairs long, the frequency that you find them in a randomly generated piece of DNA are considerably lower. So I think in the interest of time, I'm going to go ahead and stop new material right here. So I said that I was going to go to slide 10, but I'm stopping at slide 8. Okay, for the exam, one thing that I have not mentioned, but I'm mentioning now, is that you are welcome to use a single 3x5 index card. I'll make this announcement on Canvas. Yes, you can use a calculator and scratch paper. I will include pictures on the exam. I got this as a private message. Do we get a calculator and scratch paper on the exam? Are there pictures on there? Yes, I will have pictures on the exam. You can use a single 3x5 index card. I need you to scan that in, and at some point during the exam, you will be asked to show that. So on the first question, you'll be able to access that. Are we able to take it anytime Monday until 11 p.m.? Yep. 11 p.m. is the shutoff time. So I will have a ungraded assignment on our Canvas page where you're asked to submit a picture of your 3x5 index card. You can write on both sides of it. And I'm going to allow this throughout the entirety of the semester. So you'll probably have an index card that kind of you'll add to it, or you'll make a new one every exam. So that's just something you can have at your disposal. Let's see what else. The study guide that I posted, that's a great place to direct your studying. I made that for a reason, and I'm going to use that. I didn't, the reason that I made it wasn't for fun or for funsies. So that's how I'm writing the exam. There's a lot of material on here, but you can definitely do it. There will be some questions that are, I like to think about questions kind of as like low-hanging fruit questions that are like, I'll give you a structure of a ketose and I'll ask, what is this? Is this an aldose? Is this a ketose? Or I could ask, a little bit more nuance to that would be, this is an aldoketose. This is an aldohexose. I'm going to release the correct answers for the exam a couple days after. So probably by like Wednesday. Um, in addition to that, starting next week, we will begin using Top Hat, which you should see it now on your Canvas page. I would not worry about Top Hat, and please don't email me questions about Top Hat until we've actually started using it. But that's when we're going to start using it on Wednesday after this first exam. Um, let's see. I think that's pretty much it. The questions are going to be multiple choice and multiple answer. Yes, the top hat questions will be those are our participation points. The questions are multiple choice, multiple answer. The multiple answer questions are questions like, um, this molecule is an aldose. It is a hexose. It is, it has a carbonyl on carbon number 1. I got a private message. I remember you pointing out which 5 aldoses you wanted us to know, but can you go over which 3 ketoses you want us to know? Those are the 3 ketoses that are on that PowerPoint. I believe it is from, let's see, day 4. Day 4, day 4, day 4. Let me see. Yeah, okay, so here I'm going to share my screen real quick. Here are the 5 aldoses. And then, sweet, here's 3 ketoses. The ones that are boxed. Sorry, I did not say that. The ones that have boxes around them. Sure. Remember keto doctors bring fun. Nice. Great. I like it. That's clever. Okay. Well, if you have questions over the weekend, I'll respond through email. I'll do my best to respond through email. The exam starts on Monday at 9:30 a.m. You have 50 minutes, just the exact same length as our class. If you have an accommodation where you get more than 50 minutes, please send me an email reminder. That will be really helpful, and that will ensure that you get that time as opposed to any sort of. No, I mentioned this last time. It's open till 11 p.m. So it starts at 9:30 in the morning and it closes at 11 p.m. Okay, everyone. I hope you have a great day, and I hope you enjoy your weekend. And I'll see you again on Wednesday. So don't come to a Zoom. There's not a Zoom on Monday morning. Good luck on the exam, everyone.